Hello, welcome back to this bacteriology course. In this lesson, we are going to talk about a specific bacterium called Staphylococcus. Actually, there are a lot of different species of Staphylococcus, like Staphylococcus aureus, epidermidis, saprophyticus, hemolyticus, and others. This bacterium is a gram-positive bacterium, and so it has all the characteristics I told you in the first lesson about the gram-positive bacteria. It has also some specific proteins and enzymes, like protein A, which is covalently linked to the peptidoglycan, and it has an affinity to Fc receptor of immunoglobulins, and it also blocks opsonization and phagocytosis. The enzymes it produces are, for example, coagulase is a clumping factor. It's also known bound coagulase because it binds fibrinogen and it converts it to insoluble fibrin, causing Staphylococcus to clump. Other enzymes are, for example, collagen, elastin, fibronectin binding protein. Another important thing is that the bacterium is able to produce many different toxins, like cytolytic toxins, alpha, beta, gamma, delta, and pantone, valentine. Then 2 kinds of exfoliative toxins, 8 kinds of enterotoxins, and a really dangerous toxin called toxin shock syndrome toxin, which is most commonly known as TSST1. So let's first talk about the cytotoxins. The cytotoxins lyse neutrophils, causing the release of lysosomal enzymes and so the damage of surrounding tissues. For example, alpha toxin disrupts the smooth muscle in blood vessels, and it's toxic to a lot of cells like erythrocytes, hepatocytes, platelets, and other cultivated cells. It integrates to host cell membrane, causing the formation of pores, and so the efflux of potassium and the influx of sodium and calcium, and so the osmotic swelling and the cell lysis. It also causes septic shock, so it is really dangerous. Enterotoxins are also really dangerous for us. There are different kinds, different types of enterotoxins, like enterotoxin A, which is most commonly associated with disease, and enterotoxin C and D, which you can find in contaminated milk products, basically. Enterotoxin B usually causes a disease called pseudomembranous colitis, while TSST1 induces cytokine release from macrophage and T lymphocytes, and so the increasing of sensitivity to endotoxins. It also produces leakage of endothelial cells and penetrates mucosal barrier. Catalase enzyme catalyzes the conversion of toxic hydrogen peroxide to water and oxygen, while hyaluronidase hydrolyzes hyaluronic acid in a cellular matrix of connective tissue. Staphylococcal enzymes are also, for example, fibrinolysin, lipases, nucleases, penicylases, and fatty acid modifying enzymes. These enzymes are really important for the bacterium in order to be able to move inside the cells without being killed. Staphylococcus is a transient colonizer of the skin. You have this bacterium everywhere on your body. It can be transmitted with a direct contact and with fomites. It means with the contact of your infected hands and your mouth, your nose, and your eyes, essentially. So it's really important, the hand washing. Staphylococcus aureus causes a lot of different syndromes like Ritter's disease, vomiting, diarrhea, abdominal pain, so intestinal alterations. toxic shock syndrome, which is characterized by fever, macular erythematous rashes, hypotension, multi-organ involvement, and desquamation of palm and sole. It also causes cutaneous infections like impetigo, folliculitis, furuncle, carbuncle, and wound infection. The main complicancies associated with the bacterium are bacteremia, endocarditis, pneumonia, empyema, osteomyelitis, and septic arthritis. If you want to know if you have Staphylococcus in your plate in laboratory, you have to do some tests like mannitol salt agar test, and if you want to understand which kind of Staphylococcus you have to do other tests like coagulase tests that are based on the fact that some Staphylococcus produces some kinds of enzymes which are different from other kinds of Staphylococcus. One of the most important and specific characteristic of all kinds of Staphylococcus is that they are able to replicate in the presence of salt. So they are able to grow in specific plates with 0.7% of concentration of salt, and this specific field is called mannitol agar field. So this is everything for now. I'll see you in the next lesson where we'll start speaking about another kind of bacterium. So see you then. Bye!